All right, welcome everyone to the launch of the meetup of the Synthetic Biology Australasia Brisbane node, sponsored today by Decode Science and Twist, and also by New England Biolabs. First of all, SBA Brisbane acknowledges the Turrbal and Jagera people as the traditional owners of the land on which this event is taking place, and we pay our respects to their elders past, present and emerging. We recognize that this land has always been a place of teaching, research and learning. So since this is the first event of SBA Brisbane, I'd like to share with you our vision and explain to you the purpose of this organization. So we envision a highly innovative SynBio industry in Brisbane and the wider Queensland area that combines the research excellence of local universities with the vast natural resources of the region to create the next generation of world-leading bio-industries. So our purpose is to develop a Brisbane-based SynBio network as a local node of the nationally and internationally acting SBA society. Our purpose is also to promote collaborations, discussions and links between research, industry and the public, to foster scientific excellence through targeted skill development, events for researchers in industry, and to promote SynBio concepts and discuss them with the public. And we are starting with this meetup tonight. I haven't introduced myself so far. I'm Alex Gissibel. I'm a research fellow in synthetic biology at QUT, and I'm the node leader of SBA Brisbane. And I want to emphasize that we don't have a dedicated organization team here at SBA Brisbane, because we think that SBA Brisbane should be a community effort where everyone can contribute and get creative. However, I still would like to thank Laura, Juan and Marius for their help in organizing this event tonight. And if you would like to get in contact with us, I encourage you to do that. You can email us sbabrisbane@gmail.com. You can follow us on Twitter @SBABrisbane to get the latest news in SynBio for the Brisbane area. And you can also get access to our Slack channel if you want. We are going to use the Slack channel to organize and and coordinate basically the SBA Brisbane node. And but you'll need to get in contact with us to get access. And this event here and the future events are going to be are being recorded, and we'll put those recordings up on YouTube. So I would like to welcome our first speaker for tonight. That is Rob Speight. Rob is a professor of microbial biotechnology at QUT, and he's also their program leader. That's the Industrial and Synthetic Biotechnology program, and he is the chief investigator of the Center of Excellence in Synthetic Biology. So if you've turned on your microphone, then you can take it away.
Yeah, I think so. Yeah, so thank you very much, Alex, for organising tonight, and for the team. It's such a wonderful venue, and it's nice to get people together. So this event was supposed to have happened, I think, in April. And myself and Nina were lined up to do the talks then. So I've had about 6 months to practice and polish this talk. So you should expect it to be very good. I didn't honestly write it today, but we're drawing from knowledge. So also, I just want to acknowledge that this is the first event in SBA Brisbane, and I'm the Vice President of SBA at the moment. So on behalf of the SBA committee, you see the lovely people up here, including Lyra, who's gone somewhere. But yeah, on behalf of the committee, I just want to thank everyone, and it's just so exciting to see these nodes kicking off and all this kind of grassroots activity happening in synthetic biology. Just by a bit of introduction about SBA and where we came from. Thanks, Claudia, for these slides. But Claudia, who's in the audience, was very much at the genesis of SBA, and it all started relatively recently, really. So SBA is a fairly new organisation. Back in 2013, there was this workshop organised by Claudia and Colin Scott, so AIBN and CSIRO, where the community, as it was at the time, really came together to think about what we could do as a community going forward and how do we build what really wasn't existing in Australia at the time. I think you agree, Claudia, a synthetic biology community and industry. And now that workshop, so SBA was formed in 2014. Claudia was the chair of SBA, and again, Claudia very much led the building of SBA into what it is today. So even though it's quite young, SBA has achieved a lot in its short number of years, mainly around running conferences have been the big achievements, and I'm sure quite a few people in the room have been at maybe all of these conferences, or at least some of them. So back in Canberra in 2016, and then Sydney in 2017, and then most recently last year, which I think a lot of people here were at, up in Brisbane at Customs House. But what's been, I say, really awesome to see this year is the emergence of these nodes. And so in 2020, we've had Sydney in particular being very active, organising quite a lot of events. I think the first meetup they organised, which was fully online, because given, bear in mind that all of this has been happening during COVID times, I think there was 140 registrants at that first event that they had. So in some ways, this transition into online has really opened up the potential for impact from these kind of meetups and really broadened the engagement. And it's again, great to see. I think we've got about 40 people online today, as well as the 30 or so that we've got in the room. So this has really mean that there's been a lot of activity in SBA this year. Just in the bottom left-hand corner there, you see 2021. There is going to be another SBA conference in 2021. It's just starting to be planned at the moment. Looks like it's going to be in Canberra. And we're still trying to work out with the COVID emerging exactly how that's going to look, whether it's going to be online or in person, or most likely probably blended. But that has all got yet to be worked out. All right, so that's just a bit of a background on SBA, and a welcome to you all from SBA. So this is obviously a social part of the evening, and we're all having beers if we're here, and maybe at home on Zoom, maybe you're having a drink as well. So I thought we'd just start off with a little bit of a quiz. So firstly, does anybody know actually who these people are? Can anybody name anybody? Just shout out. Trevor Noah, yeah, I wouldn't have known who. So Trevor Noah is this guy in the top right. Anybody else? Yeah, Serena Williams. Who? Jaden Smith. Yeah. Maybe you're getting, see the young people in the audience are the ones who actually know some of these pop references. For the older people, anybody into rap in the 90s and early 2000s? Jay-Z? Katy Perry? Anybody? Yeah. So I'll put one more up as well. Do we know who that is? A bit different. Anybody know what links these people? I'm going to get to a synthetic biology point at some point here. Yeah, being rich is very important in what links them. unless you're really following closely the synthetic biology investment community, you might not actually know. So what actually links them is that they all invested in the recent Impossible Foods venture capital round. It's been truly amazing, actually, the pace of increase in investment in synthetic biology, obviously largely driven out of the US and California. But in times where the capital markets have seen great upheaval over this year with COVID and a lot of uncertainty, for synthetic biology companies to have raised $3 billion in only the first half of this year is really quite staggering, and shows where this industry is going and what potential it has to really transform society, and where people think the value is going to be going forward. So it's maybe a little bit small on the screen to see, but you can see there Impossible Foods are number 2 on the list with a $500 million raise this year. And although you'll see quite a lot of medical biotechnology companies up there as well, there's quite a few which are in the food space, which again is really showing how synthetic biology is transforming many aspects of our life. Pharmaceuticals has been one for a while, but food is definitely seen as one that's emerging. Who's not on that list is Zymergen. So just earlier this month, Zymergen had a raise of $300 million as well. So that's increased the number quite a bit. And that's just the companies who are raising money this year. So there's also a range of companies which are already floated on the stock market. And so you can see how investors are tracking their performance in real time. And there's some quite staggering numbers on there as well. I don't know if, Kirill, you might have invested, you're kind of savvy about these kind of things, but we'll probably use Twist to buy our DNA. Maybe if we'd invested some of our money, instead of buying their products and the shares in the company, we might be richer than we are now. But you can see from the performance there, really how companies in this space are really outperforming the market and performing very, very well. I just put Amyris for a listed company, and they've got quite a big increase there. You have to bear in mind that when they floated on the stock market about 14 years ago, they were selling shares at $20 each, so they're still not returning the value to the original investors yet. But they continue to raise money as well, and just have got an extra $200 million into their funds. So these companies who make it into the mega bucks, people like the Twist and so on, generally termed unicorns. So I just posed the question here: where do we think Australia's next unicorn might be coming from? Do we have companies here who are going to be getting billion-dollar valuations in the not too distant future? So I just listed a few of the companies that I know of in this space. A lot of them are quite young and emerging. I put your company down there as well. It's always nice for people to think, maybe what you'll do in the next step in your career might be to start a company as well and generate something with billions of dollars of valuation and benefit to society as well. We've also got companies like Louina Bio and Patheon as well, more in the biomanufacturing space, and they are companies who are both recruiting significantly at the moment. Both are going to be taking on about another 50 people in the next 6 months as their order books are growing. I just wanted to give a shout out to Inflazome as well. You might have seen that in the news this week. A company led by Matt Cooper from UQ. Also give a shout out to a friend of mine, Avril Robertson, who was one of the founding scientists of the molecule, discovered the molecule in the early work to bring that through. But Inflazome, as a UQ spin out, was just acquired by Roche on Monday for 318 million euros, so $460 million. So pretty big achievement. So these things do happen. And one of the reasons why they might happen here in Queensland is that we do have a good environment potentially for doing it. It's certainly a positive culture, a lot of it driven through the Queensland government and the Biofutures initiative. So the Queensland government does recognise that industrial biotechnology, synthetic biology is a growth area. We've probably all heard a lot from the now ex-Queensland Chief Scientist Paul Birch about the promise of synthetic biology and what it could mean. So we do have a good culture there as well. The other strategy, which does overlap with us, you might have seen, is the craft brewing strategy. Some of you are drinking from it at the moment. So it's still fermentation. I'm sure there's some angle there. I know Marius was drinking the GMO yeast-derived beer at SynBio Beta last year with a hop flavour generated inside. So maybe we can overlap there as well. I just looked at the strategy the other day, and I just pulled this picture out. Maybe we could collaborate and teach them about how to wear lab specs. I'm constantly telling undergraduates not to wear specs on their head, anti-medic. We want to protect our eyes. So I've mentioned a little bit about the economic potential of synthetic biology, but I think it's very important to recognise the humanitarian aspects as well. And really, I don't think we're doing this just for the money. And I've just posed the question there. So why do we do what we do? So a lot of us here, if not all of us, are synthetic biologists or work in this area. But why is it that we work in this area? What do we do? Why do we dedicate our lives to this kind of work? Yeah, I won't ask people to disclose that to us in the talk right now, but maybe just reflect on that. But I think a lot of the reason why we do what we do is because we see the potential for the science that we work on to make a big difference to society. And certainly that's what motivates me, the chance to work on technologies that might get scaled up, might get commercialized, might make it into the market, and not just to make money, but to make a better product that's more environmentally friendly, has less impact on the environment and society going forward. So yeah, I think we've also seen with these companies that are coming out of California and other startups as well, that it's not so much driven by the quick exit and getting rich and retiring to the beach. It's about seeing that as a vehicle to make an impact in the world. And certainly when we've heard, like at the SBA conference last year from Bolt Threads, that's the way they were talking about what they do. It's to make a difference. And that's kind of what drives us in synthetic biology at QUT as well. We want to make a difference. We want our processes to get scaled up and to make impact. And that's very much in the DNA of what drives us as a team. So the people at QUT working in synthetic biology generally sit within what is a new Centre for the Agriculture and the Bioeconomy, which started this year. It used to be called the Centre for Tropical Crops and Biocommodities before that. And within that, there's a range of programs, and there's a program called Industrial and Synthetic Biotechnology, and that's where a lot of us sit and operate as a team. I just put some web links down the bottom. If people get this presentation later, you can have a look. So for a number of years, really, we've had this culture at QUT where we do want to see what we do, as I say, move into impact, move into scale up, move into commercialization. So over a large number of years, there's been a team built up there, which can really go all the way through the value chain, from the production of biomass and working with the agriculture industry and working with waste products in that industry, through pre-treatment of that, preparation of it, and then transformation through synthetic biology and fermentation processes through to downstream processing as well. But we're also very much encouraged to work across disciplines as well. So a lot of our projects involve people in the business school and the law faculty. And in synthetic biology, that's increasingly important when we think about social license to operate and the regulatory environment as well. So we do try and bring quite a diverse team together so that it increases the chance of our projects making it through to impact. And another reason to do that as well is if you want to get through to impact, you've got to go through this kind of test cycle. I know we've probably seen these kind of pictures for synthetic biology where you've got the design, build, test cycle as well. This is a slightly different way of looking at it. We've got synthetic biology as a discovery biology, but if we can rapidly take microbial processes through fermentation, through scale up, if we can make products in sufficient quantities that we can do real field testing on them, find out how they work, we can understand the productivities and fermentations, the economic limitations, and where we should focus future developments and improvements, you can go through that loop and more rapidly get to something that might make it. And so to do that as well, you need to have facilities. And so at QUT, again, we've invested quite a lot of time and money over the recent years in being able to go all the way through those scales. So from obviously synthetic biology at the lab scale, through fermentation at lab scale, and then through to piloting. So QUT has a pilot plant that we own and operate up at Mackay in central Queensland. And there we can ferment routinely up to about 1,000 litres. And then for field testing, generally we tend to do this in partnership with other people. I'm using some examples here of things that we've actually done. So a lot of work we've done in the past has been around feed supplements, for example. We so far haven't scaled or commercialized, say, chemical manufacturing yet, so there's some examples there. I would just note as well, I've got a picture of the bananas as well. So within the center, there's a very big group working on effectively synthetic biology or modification of bananas to increase the nutritional value of those, and they're heading toward, well, they are in human trials now and getting quite close to commercialization. So I just wanted to give you, just in the last part of the talk, just a bit of a snapshot, a bit of an overview of some of the areas that we work in at QUT. I won't go into any particular scientific detail. One of the major drivers in synthetic biology is the CSIRO QUT collaboration, this synthetic biology initiative, which got Kirill in the audience up here. Hi, Kirill. So you can't see that on Zoom. So Kirill's leading this work. We've also got a really deep and growing collaboration with CSIRO more generally. So Lara, who's here, James, who's here, Marius and Raquel, who's down in Canberra. Lara and James are both fellows with this Future Science platform, and Marius and Raquel have got scholarships. So that's quite a major focus. And then, as Alex mentioned as well, QUT is a big part of the ARC Centre of Excellence in Synthetic Biology. So this was funded last year, but is going to be starting in earnest this year. We're just very close to gearing up to making things happen. And this is very much focused around microbial synthetic biology. And again, if you can see this slide from the centre, also embodies a lot of what we talked about, where we look at QUT about taking things like agricultural biomass, using synthetic biology, but to make products that have an impact in society. So just quickly, these are the chief investigators in the centre, which is led by Ian Paulson, Alexandra. So you can see from this that Queensland and Brisbane in particular have got a major role to play in the centre. So between UQ and QUT, there's 9 chief investigators. They're out of, I think, 19. So it's a big opportunity for us collaboratively in Queensland, and the way the centre's shaping up, it's got a very collaborative feel and very collaborative atmosphere. So we'll be expecting to work really across Australia, which is very exciting. And this is how it's set up. So there's 3 capabilities. So the synthetic biology research is really underpinned by systems bioengineering. So Esteban and Lars very much driving that side of it. And in terms of the themes, we've got synthetic microbial communities. So really looking at engineering communities and systems between organisms. Synthetic organelles, so engineering of the mitochondria or chloroplasts. And then neobiochemistry, which is maybe a new word which is made up during the application process, but really encompasses a lot of protein engineering and novel cofactors and so on, and really making processes much improved from what's currently available in biology. And then feeding those through, again, the centre's looking at impacts. And so I said at QUT, we bring in people from diverse disciplines. This centre does too. So in terms of industrial translation, there's people from business schools involved, people from law who can think about how to issues around the legal aspects, and then the social dimensions around freedom to, social license to operate, and so on. And all in the end, looking to get big impact. So the science obviously is important, but also jobs, manufacturing, and showing that synthetic biology in Australia has got the potential that we're seeing around the world. So just some projects that we've been focusing on in particular. This is an advanced Queensland project called Biomanufacturing Advanced Animal Feed Supplements. And again, this kind of embodies that earlier slide that I showed you, that kind of rapid biology through scale up, through testing idea. And so this project has got a number of partners, and particularly Ridley and Bioproton. Again, hi Wendy, nice to see you here. So those products already had been developed. So there was enzymes available, so feed enzymes and probiotics that Ridley were developing. And the purpose of this grant was to really accelerate that movement through production and into improving production, and generating enough material that we can put into animals to test and to work out if they work well or not, and move them closer through the regulatory process. So there's a lot, say, work on the Bacillus probiotic that we worked on, and that was really very manufacturing focused. So there's Joe Blinko there in the lab, but we've done multiple 1,000-litre campaigns up at Mackay to make quite a lot of material for pretty large-scale feeding trials, which has shown that this probiotic is looking promising for commercialization. And then Laura was involved in this project up to the beginning of the year before she started a fellowship, and did some really impressive work around engineering of Pichia-pastoris strains to improve the production of this thermostable phytase enzyme. So again, in fact, working with Kirill a number of years ago, Bioproton developed this thermostable enzyme, which was stabilized by the addition of disulfide bonds, which were an issue for the production of that enzyme in the yeast. So you can see the new phytase and the production level in that graph there. That extra disulfide bond really was a challenge for the yeast to process and deal with, compared to the orange line, which you can see the wild type unmodified enzyme. So you can see on the right a bit of a summary of the quite extensive soil engineering that Laura went through, trying many different options to improve the production, the folding and secretion of that enzyme, and was able to get it back up to where the wild type was, to a point where it's potentially now economic for scaling. So we've done other work with feed supplements as well. Again, this isn't a synthetic biology project, but led by Lee Gebbe, who you can see up in the top right there, and James is in the audience as well, who worked on the project. And this was really a discovery project looking for novel bacillus strains and going through very detailed characterization of, say, what enzymes they make and so on, and trying really to get an understanding of what the mechanism of action of probiotics are. There's still a lot of uncertainty and debate about what probiotics work, why they work, and if they work quite often. So we're really trying to get an understanding and linking molecular mechanisms within the cell and things like enzyme production to whether they actually work in an animal or not. So it remains a complex and very interesting area to work in. So another project that we have is Waste to Profits. This is a very big project, about $14 million overall, and it's a big collaboration with about 20 partners, including UQ and several universities and research and development corporations. So it's led by MLA, Meat and Livestock Australia. So the area that we work in that is, again, in the area of feed supplements. So it's converting livestock wastes or low value streams coming out of the red meat industry and turning those into microbial feed supplements. So a big part of that for us is looking at keratin and keratin waste, so hairs and so on. So we've got one in the front row here. So his PhD is looking specifically at the protease enzymes that can degrade keratin, and it's still a fairly open question about really what drives the specificity of certain proteases to be able to break down keratin when other proteases can't. So this is actually one taking a synthetic biology approach, at least through protein engineering, using cell-free expression and so on, to test as many enzymes and variants of enzymes as possible to try and build up some understanding between the relationship between sequence and function in proteases. But otherwise, we're also looking at actual process development about how best we can use proteases and use pre-treatment of the keratins to actually generate useful products like bioactive peptides and try to characterize the bioactivity to understand the value. So a lot of this work actually came out of early work we'd done, actually driven in the fashion industry. And again, Laura was a leader on that project, where we'd recognized in textiles and fashion, the reason why a lot of them can't be recycled is because they're composites. So a lot of the clothing that we wear is mixtures of polyester and cotton, for example, or it could be wool and polyester as well. So in this case, Laura developed a process where we can take these composites, you can see in the middle picture there, the smooth strands are the polyester, and the scaly ones are the wool. So you can see the hair there, and then using an enzymatic process, we can remove those. So overall, it's the idea is being able to separate this kind of commingled material and turn it into individual products that can either be recycled or used directly as something of value. So the other livestock waste that we're working on, and this is Alex, who you met before when he introduced, and Maris's PhD project as well, is actually looking to get more value out of biogas. So anaerobic digestion is a very commonly used process for dealing with particularly the waste water streams in the livestock industry. And generally that is burnt to generate heat and power, which is a relatively low use of it. Although the generation of heat and power only tends to work at reasonably large scales. So there's a lot of plants in Australia doing small scale anaerobic digestion where that methane is just being lost or flared and the value isn't being captured. So this is work led by our collaborator at PNNL, Pacific Northwestern National Laboratory, Alex Bellier, who you can see on the left there. And the concept that he's come up with is to take that biogas, which isn't pure methane. There's a large amount of CO2, about 20–30% of CO2 is mixed in, coming out of the anaerobic digester. And so to use a methanotrope, to use that methane, but in combination with an algae or cyanobacteria, so that the cyanobacteria can use that CO2, generate oxygen, which is used by the methanotrope to grow. So overall, we end up with microbial cells derived from that gas, which we can use as a feed supplement. And where the synthetic biology comes into that is we want those cells to be as nutritious as possible. So things like elevating essential amino acids, which are limiting in animals, which otherwise have to be supplemented, for example, lysine. If we can make cells which are richer in those, then we've got a higher value microbial feed supplement. We also, kind of going back to my roots, when I was a lot younger than I am now, in biocatalysis. So we work in opiates, and I see that Andres is here somewhere. So collaboration with Sun Pharmaceuticals in Melbourne. So this is really using enzymes and protein engineering and enzymology to try and generate processes where we can rapidly interconvert between different opiates. So the poppy plant generally makes a few different compounds, and there's certain poppies which have been bred to produce high amounts of, say, morphine or thebaine poppies and so on, and it's hard to predict what the market's going to want in the timescales that the plant is grown in. So the ability to use synthetic biology and biocatalysis and enzyme transformations to rapidly interconvert between different opiate products gives a big market advantage to the companies. The other biocatalysis projects we have is we're collaborating with Colin Scott at CSIRO, and this is Raquel's PhD project, building on some really impressive work that Colin's done over the years in nanomachines. So this is where enzymes and cofactor dependent enzymes are tethered to support, and then the regeneration enzyme is also tethered to the spore, and the cofactor is also tethered to the linker between the two. So effectively, the cofactor is swinging in between the catalytic domain and the cofactor regeneration domain, giving a much more efficient system. This was published, I think, earlier this year in Nature Catalysis, which is a really exciting concept. And Raquel is working on demonstrating some of these systems and working in flow chemistry to generate some high yielding processes. So that's just a very quick, rapid kind of overview of some of the projects that we're doing. But I just wanted to finish. I've just come out of teaching for 2 hours before I arrived here, and training the future SynBiologists is a really important part of what we do at QUT, and it's certainly something that really excites me and I enjoy in my job. And what we're seeing at QUT and in other universities, I think, around the world, in Australia, is that there's a lot of new SynBio teaching being developed. There's a big demand for it, I think, from the students who are starting to realize, and when you're eating your Impossible Burger, wondering where it's come from, and can I be part of that going forward. So we are developing new teaching, particularly with the QUT brand, with a very strong industry approach, and also trying to be as innovative as well in the design of the teaching that we do here. So the picture in the bottom left is a 360-degree virtual reality video I did a few years ago at the Mackay pilot plant, and I just taught that actually this afternoon before coming here. So the students have the VR glasses on. We can't afford to fly them all up to Mackay to see our plant, so we brought the plant to them, and it really kind of deepens the experience. None of these students had ever been around a chemical plant before, even though they were kind of like second-year engineers. So to be able to see it is really, really very powerful. You can see them in the class. I wasn't today, that was a couple of years ago. And the guy who's standing up there was trying to grab stuff at the same time as well. He was interesting chat. And then also, you know, trying to bring a team together for this teaching as well. So you can see the top right corner, this was the teaching that Marius and Juan did last week. It's Maddie, who's here as well. You can't see her, she's on the other side of the screen. But you can see that both Juan and Marius were really enjoying themselves doing that teaching. I went through the whole video, but shows the best frame that showed you in a good light. So that's really all I wanted to say today. I had to, because of our funding, we have to just flick through some acknowledgement slides. And I'm very grateful for the funding that we do get to support the work that we do. But particularly, you know, this is very much a team effort. I'm not in the lab doing synthetic biology myself. I entirely rely on the team and the people who work with us at QUT, and these are some of the people here. But we've got a good environment, I think, and a really, really good team, and certainly it's a pleasure for me to work with them. So thank you very much.
Thanks a lot, Rob, for these interesting insights. We have time for a couple of questions, or maybe 3 or 4. So we are going to start with questions from the live audience here. That gives time, that will give the audience in Zoom time to post questions in the chat. So yeah, please go ahead, ask away.
I can see the chat online as well, so if anybody does want to post anything in the chat, I can see it. Excellent.
Thanks for your talk, Rob. At some point, something that Alex also mentioned was that trying to make this SynBio a community. What are like your thoughts in terms of bring the SynBio to the actual community? So we are here in a very academic environment. So what are your thoughts, like how we can bring all our audience here towards the present community in this knowledge?
Yeah, good question, Axa. And it's true, there are no normal people here in the audience today. I think we're all scientists. Yeah, good question. I mean, it would be great to see people coming to these talks and events. I don't know how we make that happen, but I certainly don't think we should be afraid afraid of that and try and open it up as much as possible. I think there is a genuine, I mentioned the teaching before, and I think there is a demand from students who are kind of like real people as well, who recognize that SynBio is interesting and important. I think when we see things like Impossible Burgers being available in the shops and the impact, people are going to wonder and want to know more. So I think we've got to be open to it. So we did go out on the street a couple of years ago and just with a video camera and just casually kind of asking people in the public if you could modify anything in biology. We're trying to get a feeling about what people knew about genome engineering at the time. So we went out and talked to people, and there were a few people who said, yeah, never do it, just don't go there. But they're in the minority. In fact, most people actually said, we just want to do something that's going to save our pets. Can you make them live forever? Can you cure diseases of dogs? And then they start crying, and really embarrassing.
All right, are there any other questions from the audience here?
There's one question online. Which sectors of industry do you see most benefiting from SynBio in the future? I think the closer you get to the core needs of society, we all need shelter, food, clean water, and so on. So food is going to be a big one, but it's complicated. Yeah, 'cause we've got agriculture. I just talked about the work we do in livestock. So we do a lot of work trying to help the livestock industry and improve the livestock industry, but that's not without its challenges as well from an environmental footprint point of view. So some of the excitement, I think, in this field is the complexity and how we can try and strive just to be overall better, but it's virtually impossible to be perfect. So I think food is a big one. I mean, chemicals already is massive. I saw some, I can't remember off the top of my head the data, but something like, I think, pushing 15% of global chemicals are now bio-based, which is something in the order of $20 billion a year or something. So these things aren't necessarily new. So industries are being affected already, but yeah, so food and chemicals are probably the big one.
All right, let's thank Rob again. And we are moving on to the second and final speaker of today. That's Dr. Nina Pollak from the University of the Sunshine Coast. She's a CSIRO Future Science Platform Fellow, and her research is focusing on the development of smart biosensors, for example, for bioremediation or synthetic biology purposes. And she's going to talk about what she's been up to the last 3 years?
Correct.
All right, take it away.
Thank you. So I guess I will start with why do I do what I do? Because Rob posed this question and I quite like it. So I came to synthetic biology from a quite different background in biochemistry, and I'm a fan of nature and environment. So I'm a keen surfer. I'm all about the environment, and I feel we have to do something about it, about the pollution in our environment. And I'm focusing on water remediation. So I thought we need to find some new technologies that can help us solve this challenging problem of pollution in the environment. And therefore, I basically started this research project, which is pretty blue sky, to make some mobile tissue engineered pseudo-organisms. So I'm a synthetic biology research fellow, and I got funding from CSIRO and the University of the Sunshine Coast. And I have a co-sponsor at the University of Queensland and also with the Genealogy Research Center. So first I would like to start and tell you actually what I think is a pseudo-organism. So let's have a look on some of the examples that are out there. So this one up on the screen is actually the example that inspired me to come up with this idea and start the project. It's a jellyfish-like pseudo-organism. And it was published in 2012 by Kevin Kidd-Parker's group from Harvard University in collaboration with Caltech. And what they did is they wanted to engineer or basically recreate the juvenile form of a jellyfish, this kind of medusa that you can see up there. And that was done by using some printer and some PDMS, just making this little form, which looks a tiny bit like a blossom. And on top, they seeded some cells. And these cells are heart cells. And they got them actually from neonatal rats. So they had to unfortunately harvest those heart cells and seed them on top. And now the interesting thing is, if you put some electrodes in and you kind of stimulate these heart cells, they will beat synchronously. And if you did everything right, it will pretty much move like a jellyfish. So here we go. This is our jellyfish-like pseudo-organism and first example of pseudo-organisms. Another example is more sophisticated. It looks a tiny bit like a stingray, I would say. So now we are getting a bit more advanced in how we build those pseudo-organisms. So you have more layers and you have even a gold skeleton in there. But the principle is still the same. It's this PDMS layer, which is not biodegradable. And then on top, you again seed heart cells, which they got from neonatal rats. Now, the interesting bit about this particular stingray is that it actually will move towards light. So if you have a look here, you will see light flashes appear. And whenever the light stimulus comes in, this stingray will actually move. And that works because the cells were modified. They have now, they express channel rhodopsin, and that's a light-gated ion channel. And so as soon as the light stimulus comes in, you have some muscle movement. And they were really good with their engineering. So they used kind of a serpentine-like pattern for seeding the cells. So the activation, the impulse goes through the whole stingray. And that's why you see these kind of muscle undulations, this wave-like pattern. So now with this light stimulus, you can actually guide this stingray through a maze, which is pretty cool if you think about it. And they do that with a torch where you have a light stimulus on the left that is less intense, and on the right it's more intense. So you get a stronger kind of movement on the right side with the stronger light impulse. And so you can kind of move this stingray through a maze. Pretty impressive, I would think. And the last one of the pseudo-organisms that I would like to introduce you is made out of pluripotent frog blastula cells, so like frog embryos. And that's quite a different approach, and that's the newest member of these pseudo-organisms out there, published just this year. So this group took an algorithm and they actually started to build now little tiny, they call it living machines or robots. And so they used this algorithm, built these little kind of round things, and used forceps as well as like microcautery equipment to actually build them and shape those little things. If you have a look down there at the last picture, you can see these blue lines, and these are the movement tracks within a Petri dish, where this kind of living machine was kind of trekking along, moving along. So if we have a closer look now, these little turquoise building blocks up there, they are not moving, they are kind of just there, they are made out of more skin-like cells. And then the ones that are flickering between red and green, they are the mobile elements. So they are contractile, they are the heart cells in this case. And so like, it's pretty slow, but it's moving along. And with this algorithm, they designed lots of these pseudo-organisms for different functions. So they can just simply move along, they can manipulate an object, or they can even transport an object. And if you want, they can actually all work together as well. So now this is all pretty inspiring work, but back when I started my project, I was really still very impressed by this jellyfish-like creature, and I thought, okay, maybe we can give this a purpose. Maybe we can use this and actually remediate aquatic environment, water. So I wanted to basically build jellyfish-like pseudo-organism 2.0, and this is kind of my vision up there. So the scaffold material should be biocompatible and degradable. So I didn't want to use PDMS because I don't think the solution should be to introduce more plastic into an aquatic environment. That made no sense to me. I wanted to achieve this with 3D bioprinting, and so I needed some material that had a high degree of batch-to-batch consistency, which is a bit of a problem. The cellular system I was thinking about, it's not too great to actually use the heart cells of poor baby rats. To be honest, what could be a better system? And one of the systems that I thought of was stem cells. You can generate an unlimited number by differentiating embryonic stem cells towards producing heart cells. What's the beauty about this approach to not actually manipulate a real jellyfish and make it a GMO jellyfish is that these pseudo-organisms do not have reproductive organs, so they have a limited lifespan, and that is an advantage for us. So they cannot spread uncontrollably, which will hopefully help with using those pseudo-organisms. For the structural design, it's still going to mimic a juvenile jellyform because that's a pretty simple form. I mean, it's not that simple, but simple enough. And for the bioremediation parts, what should we, what do I need? I need an enzyme that actually does the job, and I need to target a particular toxin. So first, when I wrote this project, and because it was available in the lab, we wanted to target cocaine with cokainesterase, but that was actually a terrible idea, I must admit, because, well, it's, hard to obtain any, cocaine, just for the fun of it. So I had to find a new target, and that was theophylline. Theophylline is used in emergency settings in the US as a bronchodilator. So if you have like an asthmatic episode, that's when theophylline comes into play. And if you use too much of it, it's highly neurotoxic. So theophylline can end up in the wastewater by basically making it through our body and not being like cleaned up with our sewage treatment system. Like a lot of pharmaceuticals, antibiotics, antidepressants, synthetic estrogen, all of this actually is not taken out by the sewage treatment system and is found in our wastewater. The enzyme that does the job is cytochrome P451A2, and that's one of the enzymes that we have in our body as well, in our livers. It's a detoxifying enzyme in those terms, and it will degrade theophylline. So now I need to have some locomotion going on, and that movement comes from the heart cells. And these heart cells are made out of, well, they are differentiated from some stem cells. But a very important bit behind all this is the kind of the sensor component. So like, what is sensing my theophylline, my toxin, and how can we activate this enzyme? So what I want is an enzyme that's not all the time expressed. It's only expressed when the toxin is present, so conditional expression. So I want to switch, and what I looked at was a microRNA switch. And this microRNA switch is well known. It was first published by Christina Smolke's group, so that's not me having this idea. So I wanted to use this switch, and what you can see here is actually the nucleus of an HAK293 cell. And you see here cytochrome P450, 1A2 in there, followed by a microRNA. And then if you see this kind of light blue area, that's going to be important. But first, let's have a little look what's happening. So this microRNA is processed by the protein drosha and then further by DISA, and it's cleaved, and it will actually starts with some gene silencing. So we'll introduce that into the RNA-inducing silencing complex, and then it will inhibit the translation of the protein. And then I have to kind of measure all this, and so I use a luciferase assay. So I detect some luminescence if I add some detection reagent to those cells. So this is the case where we have no toxin. What is happening if I would add some toxin? So let's have a closer look at this orange highlighted section. This is actually a structure switching aptamer that can recognize my toxin, theophylline. And as soon as theophylline is bound, this microRNA changes in its structure. So that's why it's a structure switching aptamer. And it basically masks, hides away this microRNA, and it's not going to be processed anymore. So no microRNA means no gene silencing, and that means a lot of my protein will be produced. So I have lots of my cytochrome P450-1A2, and if I add my detection reagent, I should have lots of signal. So this is my readout system. So I started this, and sorry about that, it's like got a bit messed up here. I tested this protocol from Christina Smolke's group, and it says generate at least 4 microRNAs to have success. So I did 5 of them, and I had some success with them. 4 of them were silencing, actually only 3 were silencing my cytochrome P450 enzyme activity to some extent. And then I looked into something that's occurring naturally. So evolution has done the job already. There's a microRNA out there that will target that enzyme, and I just found it in a database, and I wanted to use that on top of it because I thought maybe this is the smarter way and the faster way. And I just engineered this structure switching aptamer in it. Going down the line, this is terribly messed up. I chose 2 of them and compared them. So one of the protocols with Christina Smolke's group, and then the naturally occurring microRNA. And this is now the assay that's very important, and I will guide you slowly through. It's a luciferase assay measuring the activity of this enzyme. And so if we just have the enzyme present, it's 100%, or I set it to 100%, and on the x-axis you have increasing levels of theophylline. So we have a control. Control means there's no enzyme present. It's actually a luciferase as control, but that's not measured in this particular assay. It's only measuring luminescence that comes from the cytochrome P450-1A2. So now we have this microRNA without the structure switching aptamer in it. So it should not react to the toxin. So the higher the concentration gets of theophylline, nothing really happens. We have some slight variation in it. But now with the aptamer, you actually can see that we get some increase in production of enzyme. So it's about this yellow highlighted area. So we can induce enzyme production by using this toxin. If I now use the naturally occurring microRNA, you will hopefully appreciate that we have far more control by using something that evolution has designed for us and just adding an element in it. So we can actually get the cytochrome P450-1A2 levels really up to wild type levels in those terms, or to 100%. So the switch is done, and we could use that. So now it's more about what else do we need with these sensor components. So as a little summary, we had moderate control with the established protocol. We had improved control with this evolutionary designed version. Sorry. The new design is really easy to use. You just use some databases that are online, microRNA base, mirror-walk, and also mFold to get your information from. So the next bit is actually where synthetic biology needs tissue engineering. So now it's about constructing this pseudo-organism. And as I said, I'm using a 3D bioprinting approach. So it's first of all, draw a model, get a diskette model, and then translate that into some instructions that bioprinter can read, and use some bio ink and mix it with the hard cells. It sounds very straightforward, but let me tell you, it isn't. Sorry. However, we got quite far with printing some jellyfish, but we're not at the end yet. How do we do that? We get some analysis done by microscopy, for example. We can stain them. Yeah, and we analyze them from the correct markers. And we follow the usual design, build, test, and learn cycle. So a while ago, I started with very simple cells. I'm using some fibroblast cells. And that, well, took me a long time to learn how to print, because as I said, it's not as straightforward, but it worked out really well. And so we moved on. We thought like, yes, let's print with hard cells. So moving on to the hard cells, why do I actually want to use stem cells? As I said, you can generate an unlimited number of those cells, and without basically isolating cells from red cardiomyocytes, I can avoid some ethical and social impact deliberations. How do we do that? So stem cells, they are finicky to work with, but there are well-established protocols out there. So you just follow pretty much different stages of cardiac development. So you give the cells little cues and you push them through different stages. Sorry. Thank you. Much better. So we start with human embryonic stem cells. We go through mesoderm, some cardiac progenitors, and we get cardiomyocytes. And what the important bit here is you don't get 100% just cardiomyocytes. That is just simply not the case, and it is good for us. We want these Purkinje fibers, and we want the pacemaker cells. So the pacemaker cells, for example, they generate the impulses that will actually make your heart cells beat. So if you only have pure cardiomyocytes, nothing will happen, because nobody, like no cell, is actually generating your impulse there. And this is how it can look then in one of the 6 pulse. So a tissue layer can just lift up and start beating as, yeah. So now the idea was, when do we print these cells with? Well, they beat at the end. We've shown this. That's around day 15. But the question is, when can we actually 3D bioprint them? So we started with day 0 cells, because why not? Let's use just some cells that are undifferentiated, and let's see if we can print them and still push them through this program by adding some growth factors. However, let me tell you, that did not work at all. So I'm cutting this story short. We went on and tried day 5 cells, because we thought, okay, they already know which way to go. They are already cardiac progenitors. They have no other way. It's not like that they will revert back to neurons or so. So maybe this is better luck for us. And yes, we can print them. However, the story is they never started to beat. So they didn't like us being harsh with them, trying to push them through a syringe and doing all the printing. So that, unfortunately, again, did not work. So we were like, okay, what else can we do? Should we do something that everyone actually said, this is not going to work, do not try or attempt this? And I'm talking about cardiomyocytes that are actually already kind of beating. So I'm talking around day 12. They have formed tight junctions, and if I want to print them, I need to like basically disrupt them again, dissociate them, their tight junctions, without actually destroying the cells, and then print them and hoping for the best. And I put this magic head up there because we have an ingredient that we mixed into the ink, and it worked once. And it worked once. And this is where the story starts and ends, because we haven't reproduced it, but we are very hopeful that this will work again. So, what now? We have to actually, well, repeat our experiments and reproduce it. And then we have to add the bioremediate function in it. And probably at the end, we also have to adjust the whole structural design. But most likely, I will probably be running out of time, and I will not create this jellyfish with the bioremediating function. However, I would at least like to be able to create a jellyfish that actually swims and is made out of the stem cells. So in summary, we had no success printing undifferentiated cells or cardiac progenitors. We had success with these beating cardiomyocytes around day 12 and mixing in these special bioink. There are that we had to overcome with lots of different things, like the printer, for example, it's the 4th machine in the lab because we had lots of issues with it. And also the material, we were pretty hit hard by COVID because lead time for materials was suddenly far greater than usual. And then it arrived with huge shipment delays, and we had 2 weeks with a product that should have a shelf life of 3 months, sorry. But we'll keep working on it, and hopefully I can give you a success story soon in the next update. What I was also wanting to tell you about is my vision about jellyfish 3.0. I know I haven't even produced 2.0 yet, but you can dream, right? So what cellular system would I like to use in the future? I would say one that is better adapted to its aquatic environment. So I would like to work with jellyfish-like stem cells. The rest is pretty much the same, but what I envision is whenever the toxin is around and the enzyme is active, I want something visible, a visible cue. Hey, it's not safe. The water is right now toxic, and this jellyfish-like thing is doing its job. So it would be really cool if it would be bioluminescent while it's doing its job, giving us a signal. I'm working on making the environment safe. And if it could even move into the direction of the toxin, that would be even greater, swimming towards the source of contamination. Big dreams. As conclusions and future perspectives, well, I showed you our sensor component for this conditional expression of the enzyme that we want to build into Jellyfish 2.0. And there are little other things that we have to think about, like signal amplification. How can we make actually enough enzyme to treat, for example, water? Well, because there will be only traces of toxin in there. And I quickly mentioned this quorum sensing. How awesome would it be if this jellyfish could swim towards the source of contamination? I would like to acknowledge the University of the Sunshine Coast and my mentor Joanne McDonald up there, as well as the University of Queensland as co-sponsor, and Justin Cooper White's lab with the 3D bioprinter. Also my mentor Alicia Anderson at CSIRO and the Genealogy Research Center, and the Future Science platform from CSIRO for funding my project, and you for your attention. And the last thing that I wanted to leave up at the screen is printing from 2 weeks ago. So that's gonna just rewind so you can see me printing, and it's gonna photo cure at some point. So you will see some purple light shining, and that's how we fix everything. Thank you.
Thank you very much, Nina. That was fascinating. I think it's such a cool project. We have 2 questions from the audience. Up there, Zach, do we have a microphone? Yeah. Great.
Yeah, thanks for the great talk from what I saw, but sorry for showing up late. I was wondering, I was really curious about the last part, when you mentioned that you're using those mature columns for printing. Did you experience any clogging when you're printing?
This is the #1 issue you have when you do some 3D bioprinting, the clogging of the nozzle. Mixing in the ingredient, it helped a lot. Changing a tiny bit of the ink, it helped a lot. That's what I can say. So it really depends on the ink that you use. But yes, clogging is an issue. So you really have to nicely dissociate the cells. So if you have some cell clusters in there, you might experience clogging of the nozzle.
So yes, when you add ingredient X, that remedies it.
I wouldn't say that that's the remedy to avoid the clogging. I think that is the remedy to make the cells survive and hopefully get them beating again. So I think it's more about dissociating the cells really nicely and mixing them in and working fast. So while I can print probably for 2 or 3 hours, my cells won't survive that long. Like you're trying, like stem cells are finicky, and even if they're differentiated to heart cells, you don't want to like get them out of the media and stress them by putting them in a syringe and then extruding them with lots of pressure for like a long time. So I would say the first 2 or 3 prints, so like I'm printing here some 6 wells, these are the good ones with the good chances. Like 2 hours down the line, whatever I print is more or less, yeah, it looks pretty, but it's not going to do the job.
Thanks.
What else? Yeah. Andrews? Sorry, Andrews.
Thanks, Nina, for a great talk. So I just had a question about pseudo-organism jellyfish. So I'm just wondering, so when it bakes, how does it get the energy to constantly?
Great question. So as I mentioned, it will have a limited lifespan and it will simply run out of energy at some point. So like these frog blastola cells that I showed you in the beginning, they could actually be in a water environment for a few days without adding any nutrients, but then they will just simply die. And that might be a good thing, but yes, you want to make the system efficient. So there are lots of ideas how you could achieve this, probably by putting some cyanobacteria in and using some sunlight or so. But yeah, it would be another challenge that I have to overcome to make this work in the future.
Yes, and Andrews is the last question from the audience. Just can you see this in the chat?
Not really.
So yeah, we'll have someone read out the questions.
Yep.
Thank you so much for the talk. It was very interesting. First of all, I would like to know if your choice of the contaminant, in this case, the second choice, was because of the switch that you had done, or because of what it was first?
Yeah, that's also pretty good question. So I've chosen theophylline because this structure switching aptamer is well known and well studied, and it's actually one of the major limitations because there are not that many structure switching aptamers out there, especially ones that work on RNA level. So that was just the perfect fit in those terms. And yes, you can go through all these CLEX process and make more aptamers, but it is actually super expensive. And I got quoted a very interesting amount because I wanted to do a different toxin in the beginning, and it was like $120,000. And that was the cheap quote without licensing or anything down the track or guarantee that it will work. So it's a pretty expensive process, especially if you want to do it with RNA. DNA is a lot cheaper. So it's a major limitation of the system, finding these structure switching aptamers that could work and can actually detect your toxin of interest.
Could you move to another kind of toxin? I mean, not necessarily a rocket target with the same switch?
Not really, because it's specific to this particular small molecule. So you would need a completely different switch.
Okay, so we don't have any questions on Zoom. One question. Yeah, okay. Please go ahead.
Peter Wolf asks, what hurdles are these to scale up for this analysis?
What hurdles? Well, I think I've mentioned a few hurdles already here. First, that I have to reproduce this. I guess biggest hurdles are making a lot of these stem cells is expensive, like the differentiated heart cells. Printing them isn't that easy, so I'm not 100% sure how you would set up a big manufacturing with all printers trying to do little jellyfish. So yeah, there are lots of hurdles in those terms to overcome, but I feel like this is down the line. There are some other ones that I have to address first before I can go into production.
All right, if we don't have any more questions, let's thank Nina again. Great talk. And now, before everyone rushes off or logs out, I would like to thank our sponsors. So that's NEB. They sponsor the food here. Thanks a lot for that. And the code Science is sponsoring a $500 prize draw for Twist products, for example, gene synthesis. And our sponsors would like to say a few words. Jenny from NEB will say a few words, and please stay tuned for the lucky winner of the prize draw afterwards.
Hi everyone, I just recently joined NEB here in Brisbane, so I really just wanted to introduce myself. I think most people here are already familiar with NEB and the products that they've been manufacturing for a number of years. And I think one thing that's important to note with NEB is that they're not just an enzyme company, but they've continued to drive innovation over the past 45 years. So particularly in supporting the synthetic biology community. Obviously the base of traditional cloning, and then they've moved towards your more advanced techniques, which many people here are familiar with. So your AnyBuilder, hi-fi DNA assembly, and the Golden Gate, which enables assembly of large numbers of fragments. So just finally, there has just recently been a paper put out regarding the Golden Gate assembly product where they were able to combine 35 fragments, so 35 fragment assembly. If anyone's interested in a copy of this paper, feel free to reach out. And NEB has a large number of tools and resources. It was mentioned to me earlier this evening that the NEB catalogue has been the Bible for many molecular biologists over the years. And I think that still rings true, although most of these tools are now available online. Excuse me. All right, and that's it for me. Thank you very much.
Thank you, Jenny. And now we have Peter from Decode Science. It is a bit of a technical challenge, but let's give it a try. I'm muting now my Zoom here, but we should still hear you, and everyone on Zoom should hear you, Peter.
No worries, Alex.
You can now.
Okay. Hopefully you can hear me there. Good to see some people talking about their work tonight who I've been contacting over the last few weeks. It's a pleasure to sponsor this evening. I apologize for not being there in person. I'm actually on holidays at the Gold Coast. It's my pleasure to give a $500 gift voucher for Twist products. And I don't know whether Alex has been able to do the prize draw as yet. And there's just a slide there showing a few of our products. So I know Rob gave a good overview of Twist before in terms of its share price, but the clonal genes and gene fragments are probably where we do most of our business. And most of you know that the difference between the Twist synthetic DNA and other synthetic DNA is it's produced on a silicon chip. So we get an excellent low error rate. So when you design your DNA, you can be sure that you're getting what you design. And we can use the power of that technology to create oligo pools for things like CRISPR and variant libraries for producing proteins, monoclonal antibody selection, things like that. If you'd like any further information, please get in touch with me. My details are there. Enjoy the rest of your evening. Thank you.
Thank you very much, Peter. Unfortunately, we didn't get the name for the prize draw, but who's the lucky winner for him?
So,
here we
the
go.
winner is Rebecca Walters.
Is Rebecca isn't here in the audience, is she?
No, she's online.
Okay, congratulations, Rebecca. We'll get in contact with you. And thanks everyone for attending this event, either in person or on Zoom. We are planning to do our next event probably sometime in November. Stay tuned. Yeah, see you soon.